Hi, my name is Dr. Joseph Gleeson from the University of California, San Diego, and Rady Children's Institute for Genomic Medicine. I'd like to tell you about our new study in the February 2023 issue of Nature Genetics entitled Comprehensive Multi-omic Profiling of Somatic Mutations in Malformations of Cortical Development, and introduce you to the lead authors, Changuk Chung and Xiaoxu Yang. Epilepsy is present in 4% of the population and is one of the most common brain diseases in children. Part of the evaluation is an EEG to identify an epileptic focus or short circuit within the brain. About 20% of pediatric epilepsy is considered intractable, meaning that it does not respond to typical medical treatment. And of those that do not respond, the disease can be life-threatening. In children who do not respond to medication, a brain lesion can often be identified on MRI or EEG, like this seen here on the MRI with the white arrow. These lesions are called malformations of cortical development and are often resected neurosurgically. The first step is to do brain mapping, like seen here, to identify the epileptic focus, and then perform neuropathological evaluation on the resected tissue. Oftentimes one sees dysplastic cells that may contain somatic mutations. Our lab previously identified somatic mutations in one particular type of brain lesion called hemimegaloencephaly. In that prior study, we found somatic gain-of-function mTOR pathway mutations that showed excessive signaling in the brains of these patients. We found mutations in the 3 genes here with the red asterisk on the left side, and then we returned to the brains of those patients that had them resected for seizures and found evidence of excessive mTOR signaling based upon phospho-S6 staining here in green. So that led us to the conclusion that these mutations occur postzygotically, indicated by the fertilized zygote here from germ cells from the father and the mother. And these mutant cells then spread throughout the brain, and in this case can give rise to quite substantial portion of the brain. In the current study, we recruited a new cohort, much larger than the previous, that represented diverse types of malformations of cortical development, indicated by the subtypes here identified neuropathologically. And we asked several questions. The first question was, are there genes other than mTOR pathway mutations in the MCD pathogenesis? So to do that, we created a comprehensive genomic profile from each patient with MCD and created a worldwide registry. From these 283 resected samples, we compared results between different centers and also between neuropathological results through this focal cortical dysplasia neurogenetics consortium, the members of which are listed here. Using brain sample and comparing with blood or saliva, we were able to do 3 phases. The first was a pilot screen where we targeted genes in the mTOR pathway. The second was an unbiased evaluation of potentially new candidate genes. And the third was a cohort validation phase. And after each of these phases, importantly, we performed orthogonal validation.
Hi, my name is Dr. Changuk Chung, a postdoc in Gleeson lab and a co-first author of this study. Our sequencing results have been summarized as follows. We identified a total of 69 genes in our cohort, which are color-coded from yellow to purple depending on the variant allele fraction. Additionally, we included 6 genes that have been recently reported as new MCD-associated genes in other studies, and these are indicated with gray nodes. The node size indicates the number of cases carrying mutations in the coding gene. The thickness of lines connecting nodes in a network represents confidence level of the functional association between the nodes, as determined by literature or experimental results. Because certain types of the MCD involve abnormal cell growth during brain development similar to tumors, we also identified genes associated with cancer using the COSMIC database, which is the catalog of somatic mutations in cancer. As expected, we observed many recurrently mutated known genes such as mTOR, PIK3CA, TSC2, AKT3, BRAF, and SLC35A2. There were recurrently mutated genes that were not reported before in MCD. Those were PPFIA4 and ATP2A1, including liprin alpha 4 and SERCA1 respectively. These were normally related with synaptic function or calcium dynamics respectively. In network level, interestingly, we were able to identify 4 different main functional modules that are related to mTOR MAP kinase pathway, calcium dynamics, synaptic function, and gene expression regulation. We next mapped the known and novel genes onto public single-cell RNA-seq data generated from human fetal mid-gestation stage telecephalon. The UMAP shows 21 cell types. I highlighted these 2 groups. These clusters show the highest level of expression of the novel MCD genes, especially dividing radioglia, suggesting that they may act in excitatory neuronal lineage, including dividing radioglia.
Hello, I'm Dr. Xiaoxu, a postdoc scholar in Dr. Gleeson's lab. I'm also a co-first author on this publication. The question we next have is whether the MCD mutations we detected and the new genes we found are actually causing the phenotypes in the patient brains. So we analyzed 2 brain tissues from normal controls and a total of 5 MCD cases using single-nucleus RNA sequencing. This technique can analyze the transcriptome of each individual nuclei from the brain tissues to massively show the cell type differences between control and MCD brains and the expressions of each of the genes in each of the nuclei. Here is a clustering method called UMAP showing the different cluster of cells. Each green dots are the transcription features from each sequence of nuclei from the normal brain. And the major cell types in the brain like excitatory neurons, inhibitory neurons, oligodendrocytes, astrocytes, and microglia can be clustered and plotted here. However, when we use the same cluster methods to look at the expression patterns of different MCD subtypes such as HME showing red, TSC showing purple, and FCD showing blue, they don't necessarily overlap with the normal controls, meaning that there are some cell types that are clustered differently. In some MCD-specific clusters, as highlighted in the circles, the expression of fibroblast growth factor receptors and epidermal growth factor receptors are exceptionally high, indicating these are highly associated with MCD-specific cell performance. To confirm this, we label the fibroblast growth factor receptors and epidermal growth factor receptors RNA expression on the right with RNA-scope technique, and the cells overexpressing those are pointed with white arrows. These cells almost identically overlap with the dysplasia cells on the left pointed in black arrows from H&E staining from the almost the same brain section. This result shows that the single cell clusters might just be the dysplasic cell clusters. We then ask whether there is a coincidence or a lot of the new genes we found that are mutated also enriched in this cell cluster. Interestingly, highlighted here, when we do a gene set level enrichment analysis, those genes that are actually found to be mutated and validated to be mutated in other patients that are not, particularly in these patients, show very high expression levels enriched in those dysplasic cell clusters. These results demonstrate their functions in the disease.
In conclusion, we found the 69 MCD genes, including 2 novel recurrently mutated genes, far from mTOR pathway. The 4 gene networks are likely to play important roles during brain development. The mutations in the MCD cohort are implicated in the pathophysiology of dysplasia cells, and these findings could lead to new molecular classification for MCD and ultimately to personalized therapies for epilepsy.
Thank you so much for your interest in our work. We'd like to acknowledge members of the Gleeson Laboratory that contributed, members of the Focal Cortical Dysplasia Neurogenetics Consortium listed here, Brain Somatic Mosaicism Network, and our funders.